Hello everyone. We shall now be dealing with the gastrointestinal infections that are caused by parasites. And we shall start the topic intestinal amoebiasis. Let us have a brief introduction regarding what is amoeba. Amoeba is a protozoan parasite and amoeba means change. That is, these parasites, they constantly change their shape. It is a single celled microorganism and amoeba has got a simple life cycle. That means it requires only one host to complete its life cycle. It is mostly seen in two forms, trophozoite and cyst. We will see both the forms later. Amoeba replicates by binary fission. As I earlier mentioned, it changes its shape because amoeba has got certain finger-like projections on its cell wall that is called as pseudopod, also known as false foot, which eventually helps amoeba for motility. Now, based on the habitat, these amoeba could be of two types. They could be either intestinal amoeba or free-living amoeba. Free-living amoeba mostly causes CNS infections and hence it would be dealt later, while the intestinal amoeba could be either pathogenic as well as it could be commensal organism. The most common important pathogenic intestinal amoeba is Entamoeba histolytica. It is the most common pathogenic intestinal amoeba. There are many species of Entamoeba, but mostly they are commensals. Also, there are these three species of Entamoeba which cannot be differentiated from Entamoeba histolytica morphologically. But yes, there are other methods with the help of which it could be differentiated. And these species are Entamoeba dispar, Entamoeba moszkowski and Entamoeba bangladeshi. They are not known to cause any infection except moszkowski and bangladeshi are reported to be associated with infantile diarrhea. Now let us see regarding the epidemiology of amoebiasis. Amoebiasis is worldwide in distribution and is considered to be amongst the neglected tropical diseases globally. 50 million infections of intestinal amoebiasis are reported globally with more than 1,10,000 deaths. And the highest burden of this disease occurs in tropical and subtropical countries like China, Central America, South America and Indian subcontinents. The infective form of this amoebiasis is the mature cyst of Entamoeba histolytica. The source is water and food that is contaminated with the cysts, and in most of the cases, that is in 90% of the cases, the host become asymptomatic cyst passer, and in very few cases, that is only in 10% of the cases does the disease progress. Entamoeba histolytica exists in three stages: trophozoite, pre-cyst and a complete mature cyst. Now let us have a look at the morphology of each of them. Here is the trophozoite. The trophozoite is the invasive form, also called as the vegetative form. This trophozoite, as you can see here, is around 15 to 20 micrometer in size and it contains a single nucleus. You can see it has got these finger-like projections that are called as pseudopodia, which are the organ of locomotion for this trophozoite, which helps in its motility. While in pre-cyst, they are the intermediate stage between the trophozoite and cyst. It could be uninucleate or binucleate. As you can see here, the picture of E. histolytica trophozoite. The cyst of Entamoeba histolytica is the diagnostic form of the parasite which is found in the feces of carriers as well as the patients with active disease. This cyst measures around 12 to 15 micrometer in size and it could have 1 to 4 number of nucleus. As you can see in the picture, it could be a uninucleate cyst, a binucleate cyst, or a fully matured quadrinucleate cyst. This quadrinucleate cyst is the infective form of the parasite. The picture shown above is the distribution of the forms of Entamoeba histolytica based on the consistency of the stool. The cysts are seen to be more in formed stools, while the trophozoite form is seen to be more in watery stools. You can see the picture of a mature quadrinucleated cyst of Entamoeba histolytica. This is the iodine mount picture of the cyst. As I have already mentioned, the mature quadrinucleated cyst is the infective form of Entamoeba histolytica, and this cyst can remain in moist stool for as many as 10 days. And one of the most important feature of this cyst is it resists chlorination, because of which when this cyst enters into the stomach, it bypasses the action of gastric juices and directly enters the small intestine without getting destroyed. Source of this infection could be contaminated water, food, and also it could be at times, rarely, by the vectors. Like flies, they may mechanically transmit the cyst from the feces and might contaminate the food and water. And the mode of transmission, the most common mode of transmission is the feco-oral route. Now we shall see how the life cycle of Entamoeba histolytica works. As you can see in the picture here, once the host gets infected with contaminated food or water that has been contaminated with the cyst of E. histolytica, and once the host ingests the cyst, this cyst then enters into the stomach, it bypasses the juices and eventually enters the small intestine. This cyst then converts to the vegetative state, that is, it forms trophozoites, and in most of the cases these trophozoites undergo encystation, that is, the vegetative state is converted to cystic stage and the cyst is then passed into the feces. 90% of the patients are asymptomatic cyst passers, but in few cases, as you can see here, these trophozoites can multiply by binary fission, they can adhere to the colonic mucosa and infect the intestine, leading to the disease. You can see the picture over here, the cyst and the trophozoite. We will have a look on the life cycle in detail. Cyst, once ingested, it enters to the stomach, it bypasses the action of gastric juices. As I already told you, it is resistant to chlorination and it enters the small intestine. Once the cyst enters the small intestine, excistation takes place in the small intestine. That means the cyst wall get ruptures and the metacysts inside it are released, forming metacystic trophozoites. There are 4 nucleus inside the cyst and hence 4 metacystic trophozoites are formed. Now these metacystic trophozoites, they are carried to the large intestine, mainly to the ileocecal region, and these trophozoites then multiply by binary fission and they start colonizing on the intestinal mucosa. Now once these trophozoites are colonized on the intestinal mucosa, they might take different courses. As for one, the most common course of this trophozoite is it undergoes encystation and do not cause any lesion. They transform into the cyst and the cyst is then excreted in the feces. causing the host to be an asymptomatic cyst passer. Then in some individuals, these trophozoites get adheres to the intestine mucosa and can produce different conditions like ulcer, dysentery, etc. And very rarely these trophozoites, they can invade the intestinal mucosa and gain its access to the portal veins and migrate to the extraintestinal sites, the most common site being the liver, and causes extraintestinal lesions. Once again we shall see the same thing, the development of the forms in the life cycle. This is the mature cyst. Once it enters inside, it bypasses the stomach and enters the small intestine, wherein existment takes place and trophozoites are released. These trophozoites on the large intestine, they adhere and start multiplying by binary fission. They might undergo encystment, form cyst, and the cysts are passed in the feces, or else this trophozoite could adhere to the intestinal mucosa and lead to the diseases. We shall see the steps of pathogenesis of intestinal amoebiasis. As now we already know, the mature quadrinucleated cyst enters through the oral route and the cyst undergoes excistation in the small intestine, thereby bypassing the gastric juices of the stomach, and once it undergoes excistation, the trophozoites are formed. These trophozoites, they multiply by binary fission, and once the trophozoites are colonized, they start adhering to the intestinal mucosa, and with the help of virulence factors of trophozoite, they start destroying the tissue of the intestine, leading to ulceration of the intestine. A very typical ulcer is formed because of this amoeba, that is called as the flask shape ulcer. You can see in the picture, flask shape, that is narrow at the top and broad at the bottom, hence leading to intestinal ulceration. Also, these amoeba can then invade the large intestinal wall, which can then migrate to extraintestinal sites, and finally can cause intestinal complications. As you can see here, we shall talk something regarding the virulence factors of trophozoite. This is the entamoeba trophozoite with the phagocytosed red cells. It has got some virulence factors like the lectin antigen, the amoebic lectin antigen. It helps in adhesion to the epithelial cell of the large intestine. It has also got amoeba pore. This amoeba pore, it forms pores on the target cell membrane, which causes the leakage of the cell contents and hence leading to cell death and phagocytosis of the cell. Also, it has got certain enzymes like proteases, which causes tissue lysis. These all actions together eventually leads to ulceration and destruction of the tissue of the colon. We shall see the course of amoebiasis. The E. histolytica once enters into the stomach, bypass the juices and enters the small intestine. Most of the cases, in 90% of the cases, the infection is self-limiting. It is only asymptomatic, while in 10% of the disease it could be invasive, that is the E. histolytica could invade the intestine and cause ulceration and dysentery, while only in less than 1% of the cases it can cause extraintestinal infection, and the most common extraintestinal infection is the amoebic liver abscess. followed by either pleural or pericardial infusions, and it can also lead to brain infections. Now we shall see regarding the clinical syndromes or the clinical manifestations of amoebiasis. As I have already mentioned, 90% of the cases are carrier state, that is, they only pass the cyst, making it infectious to other host. Then 10% of the cases could be intestinal amoebiasis and less than 1% extraintestinal amoebiasis. In the intestinal amoebiasis, as we already saw earlier, the virulence factors of the trophozoites, once they multiply by binary fission, they start causing localized tissue destruction, and hence it might lead to amoebic dysentery. Dysentery is basically bloody stools. And in amoebic dysentery, the symptoms are abdominal pain, cramps, colitis with diarrhea and bloody stools. While extraintestinal manifestations are seen by some systematic signs, which depends on what site has been involved, and the most common extraintestinal site that is involved is the liver. Now remember, when we talk about dysentery, we have already talked regarding Shigella dysentery also. So we will see some differences between what is amoebic dysentery and Shigella dysentery. So some pathological differences of the manifestation of dysentery, that is amoebic and bacillary. The ulcer in amoebic dysentery is deep, while in bacillary it is shallow. As you have already seen, the ulcer is flask shape. The margins in amoebic dysentery are ragged, and in bacillary they are uniform. A very important finding is that the intervening mucosa, that is the mucosa between the ulcers, is very normal in amoebic dysentery, while in bacillary dysentery it is inflamed. So these were the pathological differences. We shall now see the macroscopic features of the stool. The number of motions in amoebic dysentery is 6 to 10, while in bacillary dysentery it is more than 10. The amount is copious in amoebic and very small quantity in bacillary. The color of the stool is dark red in amoebic and bright red in bacillary. Again, a very important finding, the stool is very offensive in case of amoebic dysentery and it is odorless in case of bacillary dysentery. And the last differences, that are the microscopic features of the stool. The red blood cells in amoebic dysentery are seen in clumps, while it is discrete in case of bacillary dysentery. There are very few pus cells in amoebic and there are numerous pus cells in bacillary dysentery, as it is caused by a bacteria. There are very few macrophages in amoebic and numerous in bacillary. Eosinophils are present in amoebic dysentery, but they are absent in bacillary dysentery. Again, a very important finding, Charcot-Leyden crystal are present in amoebic dysentery while absent in bacillary dysentery. We shall see the invasion of E. histolytica into the large intestine. As you can see here, the layers of the large intestine: mucosa, submucosa, muscularis mucosa, the muscle layer and subserosa. Primarily, the amoeba invades via the crypts and forms the primary ulcer, which is typically called as the flask shaped ulcer. It is narrow at the top and broad at the bottom. The size of this flask shaped ulcer could be from a pinhead to few inches. This ulcer might then extend into the muscularis mucosa, and then there could be formation of sinuses. Once it enters the muscularis mucosa, the lateral extension of it might cause lytic necrosis, and this abscess may then coalesce and form sinuses. And finally, it could also pierce down the muscularis mucosa and submucosa directly or via using the blood vessels. You can see the picture of amoebic ulcer. Very nicely shown here, the flask shaped ulcer. As you can see here, it extends laterally at the bottom. A histopathological picture of a flask shaped ulcer. You can see here, it depicts the picture of a flask. Now, as the amoeba invades the large intestine, it starts causing complications. As for example, it might perforate the intestine and cause peritonitis and hemorrhage. It might cause fulminant amoebic colitis, which results from generalized necrotic involvement of the entire large intestine. It might cause secondary infection to other organs, and also a diffuse pseudotumor like mass of granulomatous tissue could be found in the rectosigmoidal region, which is called as amoeboma. And because of the amoeboma, there could be obstruction of the large intestine also. Also, there could be toxic megacolon and intussusception, and it might invade the blood vessels also. Now, seeing the extraintestinal spread of amoeba, amoeba could directly invade the large intestine and could directly extend to the skin, causing cutaneous amoebiasis, which is manifested by ulcer having irregular margins and necrotic floor. Also, the secondary invasion via the portal vein is into the liver. The most common extraintestinal site of amoeba is the liver, where the amoeba enters into the cells of the liver and forms abscess. As you can see here, the picture of amoebic liver abscess, the gross picture of amoebic liver abscess. Again, a picture of amoebic liver abscess, wherein the abscess is being aspirated for diagnostic as well as therapeutic purpose. This is the color of the pus that has been aspirated from amoebic liver abscess. It is very popularly known as the anchovy sauce pus. It is thick and chocolate colored. As it resembles the anchovy sauce, it has been named as the anchovy sauce pus. You can see the picture of Amoebiasis cutis. Once again, Amoebiasis cutis at the anal region. Amoebic meningitis, a secondary invasion into the brain. Now let us see the laboratory diagnosis of amoebiasis. The mainstay of diagnosis of intestinal amoebiasis is the stool examination. Minimum 3 samples of stools are required to be collected on alternate day because amoeba sheds intermittently, and hence 3 stools are minimum required. While the cyst forms are seen in both, that is in carriers also and in active disease also, but the trophozoite form will be seen only in active disease. It can also be diagnosed by histology, and a very lesser known method is the stool culture. Then it can also be detected by coproantigen, that is the detection of antigen in the stool. There is a commercially available immunochromatographic test, that is the triage parasite panel, wherein it simultaneously detects 3 parasitic antigen in the stool, and those 3 parasitic antigens are Giardia, Entamoeba histolytica and Cryptosporidium. There are serology as well as molecular methods available also for the diagnosis of Entamoeba histolytica. And for the examination of extraintestinal amoebiasis, The mainstay is again the microscopic examination, but in this case the microscopic examination has to be done of the aspirate, the aspirate that has been aspirated from the liver abscess. And the other diagnostic methods available are the imaging techniques. The microscopic examination of the stool is done using normal saline and iodine mount, and we try to look for both the forms, that is the trophozoite and the mature cyst. As seen here, the trophozoite are around 15 to 20 micrometer in size with having a single nucleus, and the cysts are 10 to 15 micrometer in size and they could have 1 to 4 nucleus. These are the pictures of the cyst in iodine mount and the trophozoite in the staining. The trophozoites, they are better appreciated in saline mount than the iodine mount, because the iodine will kill the trophozoites and the motility of the trophozoite cannot be appreciated, and hence trophozoites, they should never be reported based on the iodine mount. While the internal structure of the cyst is clearly appreciated by the iodine mount than saline mount, and it is even better appreciated on staining. These are the charcoal laden crystal, which are the characteristic microscopic feature of amoebic dysentery. Charcoal laden crystal are formed because of the breakdown of eosinophils. And for the extraintestinal amoebiasis, as we have already seen, imaging techniques are more helpful, like X-ray, CT scan, MRI and sonography. Aspiration of the amoebic liver abscess is useful diagnostically as well as therapeutically. Diagnostically, microscopy of the aspirated pus is done, and the microscopic observations shows trophozoites of Entamoeba histolytica and never the cyst, and these trophozoites may be only found in the last portion of the aspirated material, because it is never present in the necrotic debris which is obtained from the center of the abscess. Thank you.